Okay, so thanks everyone for joining us. Today we will be having our webinar here, Sniffing for Skunks, How Conservation Canines Are Helping Island Spotted Skunk. So everyone should be muted as you've come in, but if you're not, please just mute yourself through our presentation. If you have any questions, feel free to put those in the chat, and we'll be monitoring that chat throughout the presentation. And without further ado, I'll get us started. So my name is Crystal Charlo Schaefer. I am the Director of Development here at Working Dogs for Conservation. And our guest speakers today will be Lara Brenner from the Nature Conservancy and Mark Vick with Working Dogs for Conservation. So I'll just be starting off with our agenda. I'll talk a little bit about Working Dogs for Conservation, or WD4C, and then I'll hand it off to Lara, where she'll then share about island spotted skunk conservation, a little bit about that species, some challenges they're facing, as well as our current research. And then she'll hand it off to Mark Vick, and he'll be sharing about conservation canines and how they are a wonderful method for the research needed, and then also project details and results. And then Lara will then be sharing about a little bit more on the results and future plans, and we'll have time for questions at the end. So a little bit about us here, Working Dogs for Conservation. We are a conservation-based organization that rescues dogs, trains them to use their noses to help save wildlife and wild places around the world. We love to say that we do it to save the world. The dogs do it for the love of a ball. So this is a really, truly a game for the dogs. It's work for them, and they really enjoy it and love it. We are a nonprofit organization. We're based out of Missoula, Montana, and we work throughout the United States and internationally as well. We were started 25 years ago by four awesome, amazing women, and I have them pictured here on the slide. These are our four co-founders. And Working Dogs for Conservation is one of the largest and longest standing conservation canine organizations in the US today. So our team collectively has experience in 45 states, 36 countries, and over 100 scent targets. And most of those scent targets are new or novel projects. So we proudly are helping to lead the field. We also have a really big focus on rescue dogs. So the dogs that we use for our programming are dogs that are often hard to place in homes. They are really high energy, toy driven, almost toy obsessed. And that is what makes a wonderful working dog for us. So our focus is on rescue, rehome, and career change dogs. And then additionally, we have a great focus on partnership and collaboration. And this webinar is a great example of that. So we're partnering with the Nature Conservancy on this work. And then we are also open source. So we really have a focus on sharing this information, not only at conferences and in publications, but with the public as well and other partners. It's for the greater good of conservation that we share our knowledge and share what we learned and continue on with that. And then a little bit just about our work. So the project we'll be learning about today, Skunk work, fits into ecological monitoring. We have four focus pillars of our conservation work. So we have ecological monitoring, which you'll learn about today, but that's basically tracking with wildlife, figuring out, measuring populations, getting natural history information, data collection, all things related to that. Another pillar we have is biosecurity, and that is where we're looking for invasive species and even wildlife diseases. We have another pillar about stopping wildlife crime, and this is like wildlife law enforcement. So if you think about tracking down poachers and interrupting wildlife trafficking. And then we also have an environmental justice pillar, and our most recent project on that has included contaminant monitoring. So we utilize the dogs, they're using their noses to help us with all of those. And those are the four focus pillars of our conservation work. but that work is absolutely supported, and the foundation of it is our dog life program. So that includes intake and sourcing of our dogs, training, fitness and conditioning, all those things related throughout their life and their career, all the way through their retirement. And so all of those things together help make up Working Dogs for Conservation. So without further ado, I'm going to hand it off to Lara, and she will share more about this work.
All right, thanks so much, Crystal. My name's Lara Brenner. I work for the Nature Conservancy here in California. And our mission is to conserve the lands and waters on which all life depends. And so we work with a lot of partners around the world, including other amazing nonprofits like Working Dogs for Conservation, to advance that mission. Here in the US, the Nature Conservancy owns a number of preserves. We also we kind of serve as a land trust. We also purchase land that's ecologically important and set it aside for conservation in perpetuity. But we also do a lot of other things like cutting edge research, policy, and all the things that are needed to help protect our planet. So I'm just going to talk a little bit definitely about this project and how we came to work with Working Dogs for Conservation, but I wanted to zoom out a little bit and talk a little bit about island spotted skunks, islands in general, why they're important for conservation, and why we've decided to focus our time and energy on learning more about this really distinctive, unique island endemic species. So the Nature Conservancy is invested in islands around the world, and that's because they're really important for conservation. It might seem a little bit counterintuitive because islands are only 5% of the Earth's landmass. So compared to things like rainforest, tundra, other types of ecosystems, they're a really small percentage of our lands. But within that 5% contains 19% of bird biodiversity, 41% of threatened land vertebrates, and unfortunately, 75% of vertebrate extinctions. So not only are islands hotspots of biodiversity, but they're also hotspots of extinction. These are really unique places that are very vulnerable, isolated, and can easily experience massive changes due to, the climate changing and due to other human threats. Next slide, please. So I work primarily in the California Channel Islands, which are an archipelago of 8 islands off the coast of Southern California. These are the 4 northern islands here. From east to west, you have this little sliver, Anacapa. The big one that looks kind of like a rubber chicken is Santa Cruz Island. The next one is Santa Rosa Island, and the farthest one out is San Miguel Island. And these islands have never been connected to the mainland, so they are hotspots of endemism, meaning species that are found nowhere else on Earth can be found in this archipelago. They've got, in addition to a lot of terrestrial biodiversity, they're bathed by these. You can kind of see it in this aerial imagery here, of mingling of cold and warm water currents. So there's a lot of marine diversity as well, an abundance of marine life, and they're a really special place to work. People call them the Galapagos of North America, and the biodiversity is kind of comparable to the biodiversity that Charles Darwin encountered when he visited those islands hundreds of years ago. These islands have been inhabited by people for 10,000 years. Actually, some of the earliest sites of human habitation in the Americas can be found on the Channel Islands. And in the last 200 years or so, with European settlement and colonization, they've experienced a lot of rapid change, destruction, and more recently, recovery. Next slide, please. So like I mentioned, the Chumash people of Southern California lived on these islands for 10,000 years. Starting about 200 years ago, they and the other island inhabitants, like the island fox and the island spotted skunk, experienced a lot of really rapid change. So there was overhunting and overexploitation of the resources, including sea otters, which were driven to extinction on the islands. Pinnipeds were hunted, seabird eggs were hunted. In addition to that, ranchers introduced grazing animals to the islands in huge numbers, primarily sheep, but also cattle. Deer and elk were introduced as hunting opportunities. And these islands had never experienced grazing at that intensity before. And within a couple decades, all of the amazing, unique plant life had been really challenged by these invasive grazers. And in some cases, some plants went extinct. Others became so rare that they were basically undetectable. And the islands themselves kind of transformed from this lush ecosystem of chaparral and coastal sage scrub into but basically like a moonscape. While that was happening, there was also a lot of contamination of the oceans. There was a DDT factory just off the coast of Southern California that was spewing DDT directly into the ocean. Many of you might be familiar with the story, Rachel Carson's book, Silent Spring, and the story of how DDT cratered bird populations by thinning their eggshells. So the Channel Islands were, this was an effect around the world wherever DDT was used, but the Channel Islands were especially affected because of the proximity of the factory that was producing it. So bald eagles, peregrine falcons, and other raptors vanished from the islands. And even today, those long-lasting impacts are still seen in the marine ecosystem. Pinnipeds and dolphins still have a lot of DDT in their tissues. So basically, the islands were really hurting. Next slide, please. So luckily today, starting in about the 1980s, we kind of entered what we call now the restoration era after the exploitation era. So all of the islands now are set aside for conservation in some form or another. There's a mixture of land managers. The national park owns 5 of the 8 islands. TNC owns part of Santa Cruz Island, which is in light green. It's the Santa Cruz Island Preserve. The Navy manages and owns San Nicolas and San Clemente Island and sets aside large chunks of those for conservation purposes. And then Santa Catalina Island is in a land trust as well. And during that time, the land managers have worked to take the ungulates, the grazers, the sheep, the pig, the goats off the islands, let the landscape and habitat recover, and let some of the endemic, unique species come back. And we've had so many species delisted, taken off the endangered species list since that's happened. The most maybe iconic is the island fox, which was impacted by interactions between all these invasive species that were introduced. It's a really amazing story. It's very complicated. Our focus is on skunks today, so I don't want to go into the fox rabbit hole, but if you ever need a pick-me-up, you can read about the island fox's story. The Friends of the Island Fox have a great website where you can learn all about the amazing effort that recovery took. In addition to island foxes, we had a number of plants come off the endangered species list. Even just as recently as 2023, we had 2 plants removed from the list because they'd recovered in the absence of grazers. So I'm going to kind of focus in now on Santa Cruz Island, which is the island that I work mostly on. And this island has 12 endemic species that are found nowhere else on Earth, including the Santa Cruz Island scrub jay, which is the only single island endemic land bird. Channel Islands tree poppy is found there, Santa Cruz Island fox, like I mentioned, and the island spotted skunk, which is found on Santa Cruz and Santa Rosa Islands. Next slide, please. So with that context, I'll dive a little bit more deeply into this particular conservation problem surrounding the island spotted skunk. So a little bit of background about this particular species and the subspecies that's found out on the islands. It's only found on Santa Cruz and Santa Rosa Island and nowhere else on Earth. We don't actually know how many there are, but we estimate it's fewer than 1,000 individuals. Their closest relative is the mainland western spotted skunk, and island spotted skunks first arrived out on the Channel Islands somewhere between 10 to 13,000 years ago. The northern islands actually at this point were all connected into a super island called Santa Rosa. Sea levels were lower, and the mainland was only 6 miles away. Everything that is on the Channel Islands today either swam out there on its own power, flew out there, was washed over in vegetation or other floating debris, or was brought by people. We don't know how island spotted skunks got out there. They probably didn't fly, but they may have washed out on a log where they were making their den, or the timeline also kind of lines up around the time when people were first exploring the island. So it's possible they were brought over intentionally or as a stowaway. Next slide. So a little bit of background about how we started to focus on island spotted skunks. I mentioned briefly the story about the fox decline and recovery, and this chart kind of shows how quickly that all happened. So starting in the 1990s, we had researchers on the Channel Islands studying the fox population, and they started to notice that their capture rates were declining really quickly. And in the course of 10 years, or maybe 20 years, we went from a population of around 2,000 foxes on Santa Cruz Island down to only 100 in the late 1990s. And on San Miguel and Santa Rosa, it was even lower. There were only 15 in the wild. And this started this big flurry of activity to try to rescue the species from extinction. So the foxes, all the foxes that were remaining in the wild, were brought into captive breeding. They were put on the endangered species list. A lot of funding and energy went into figuring out the causes of the decline and trying to remove the threats from the landscape. And so during this time, TNC, the Nature Conservancy, and the Park Service were taking pigs off the island. We were removing golden eagles from the island and releasing them on the mainland, restoring bald eagles and captive breeding foxes, and just trying to put the landscape back together. Golden eagles had been predating on foxes, and once they were removed from the landscape and replaced with their native counterpart, the bald eagle, we kind of deemed the island safe for foxes to live again. And so they were all released from captive breeding, and around 2008, the captive breeding program ended, and the fox population, which is shown in this orange line here, started to recover. Now, interestingly, we were trying to trap foxes and learn about their populations, but skunks also go into the same fox traps that we set out to try to monitor fox populations. And so, almost as an accident, we were also measuring kind of a proxy of the population of how the skunks were doing. And what we found was that as the fox captures declined, this orange line here, skunk captures just skyrocketed. They went through the roof. We went from 1 to 3% capture success to almost 40% trap success. So for context, that means we went from catching one skunk per 100 traps to like 40 skunks per 100 traps. And then sure enough, as the foxes started to recover, our skunk trap success declined as well. And you get this really kind of perfect seesaw relationship between the two species. And now they've kind of been hovering around a 1 to 3% trap success. And we kind of realized at this point that this is a species that's, just as unique and interesting as the island fox, and we don't really know very much about it and what might be threatening its populations. So next slide. So we were wondering, why have skunk captures gone down? So we had a bunch of questions. It obviously seems like this is something the foxes and the skunks have some kind of a relationship. Are they competitors? Do they compete for habitat and limited food or other resources? Are skunks avoiding foxes as either a predator or an aggressor? So are they avoiding where we're trapping foxes because they don't want to encounter them? Will the skunk population stabilize and will it remain viable in the presence of foxes? Obviously, these species coexisted for several thousands of years, but will climate change kind of throw a wrench in the works and change this relationship by altering the resources that are available? Oh, next slide, please. So once the foxes were kind of in a good place and secure, we turned our attention to the island spotted skunks, which had not received a lot of attention up to that point. And one of our first acts was to form the Island Spotted Skunk Working Group, which was a big group of managers, researchers, government agencies who all might have a stakeholder interest in this species. And we kind of developed our major goals for monitoring, for research, and for management. Based on what we determined from that group, which you can also read about the Island Spotted Skunk Management Plan, which is available on the Island Spotted Skunk website, we decided we want to continue to monitor long-term skunk trends through our Fox live trapping grids, which were not set up for skunks, but they have been going on pretty consistently for several decades. We put radio collars out on skunks to monitor their use of habitat, their territory size, and their survival over time. We set out a network of camera traps to kind of monitor the same, but at a higher intensity. And then anytime we did catch a skunk, we collected every kind of biological sample we could, including whiskers, which can tell us about their diet over a long period of time, microbiome information, scat if available, and ectoparasites. And this can all give us information about genetics, disease, other risk, diet. Yeah, we just had a lot of questions about skunks that we needed to answer. Next slide, please. So we've learned a lot from these efforts, and there's again a lot of information about this on islandspotted.skunk.com. The summaries from some of these projects are available, but we ran into a lot of challenges that are kind of unique to the island spotted skunks when you compare it to our experience working with foxes, which are in some ways a lot easier. We don't catch very many live skunks, so every year we catch about 200 foxes, so lots of opportunities to put out collars, collect data, that kind of thing. We might catch on Santa Cruz Island between 1 to 10 skunks. So very limited opportunity to collect a meaningful amount of data. They're really challenging to collar. They live in dens, and so they're really hard on the collar. They rip the antenna off. They're small, so the collars only last for a few months at a time. So again, not a lot of opportunity to collect data. We found that the camera traps are probably the most cost-effective way to collect data on skunks, but obviously, all you get from that is a picture. You can say a skunk was here, but you don't know if you can't identify the individual, you can't get to a population estimate, and obviously you can't collect any genetic or other bio data. And then the other issue is nowadays foxes are very abundant and they're very curious. They like to get into everything. And so we don't have a lot of opportunity for non-invasive, non-targeted data collection like hair snares. It would just get swamped out by fox activity. So we kind of wanted to get, we know we want a population estimate for the skunks, which you can get from genetic information. We want to know about their diet and how that impacts their relationship with the fox, how that changes over time, and how their dietary niches overlap. And we want to know about disease threats. And we're having a hard time getting that kind of data through our existing methods. Next slide. And so that's when we kind of turn to working dogs for conservation to probe this idea of whether we could train a detection dog to help us find spotted skunk dens and scat. And so with that, I will pass it over to Mark to talk a little bit about the process of selecting a dog, training a dog, and then our pilot study on the island.
Thank you, Laura and Crystal. Hi, my name is Mark. I've been with WD4C about 12 years, the last 4 of those as the Assistant Director of Programs. I had the privilege of working with Moose for this exciting pilot project. Moose is a 2.5-year-old Spaniel Aussie mix. He was rescued in Montana. This was his first field deployment. One of his favorite toys is a squeaky Kong ball. And as you can see here in the picture, he is fully embodying his role as a skunk detection dog, or maybe a doppelganger. Next slide. When Laura and I first started talking about the possibility for utilizing detection dogs to locate skunk scat, one of the things that resonated with me was approaching the training methodologies from a different perspective than how we typically locate scat. Rather than focusing on or training on locating the scat, we wanted to focus instead on teaching moose to locate dens occupied with skunks. And this idea was based on knowledge and experience I had gained on a previous project back in 2015, where dog teams were trained to focus locating dens occupied with eastern striped skunks. This was on the mainland in West Virginia, that past project. So we hypothesized that our best chance of success was to train moose to locate dens with live skunks and train moose to locate skunk scat as a secondary target at and around the den sites. In general, skunk scat can be pretty fragile and often concealed at depth in dens. Next slide, I think. Yeah. So with that background information in mind, Laura and her team were able to collect skunk hair and bedding from some of the Santa Cruz skunks, like she mentioned in the traps. We also obtained hair, bedding, and scat from a few different close cousins, the western spotted skunks residing at Sequoia Park Zoo and Turtle Bay Exploration Park in California. With those training samples, we focused the setup and the context to mimic places resembling dens, burrows, and all sorts of skunk style houses. So when Moose is searching in the landscape, his expectation is to search the landscape for those target odors, but to prioritize his energy and efforts in a way that would be ideal for locating dens. So this approach supports coverage of larger areas while conserving his energy by not focusing on just looking for the scat in the landscape. The den scent picture is a much more accessible odor for moose as well. I think we can go to the next slide. Or maybe not, but we can stay here. So we layered on scat as a secondary value target so that if he did encounter scat outside of the dens, it would have value and he would still tell us about it. But his primary focus was occupied dens. Also, once at the dens, he had an expectation to shift into, okay, now I'm at the den. My next job is to point out any nearby scats. There were some instances where we found scat just outside the den. So it was really helpful once we got into an area with fresh activity that Moose could also help us pinpoint where the individual scat was. And to be able to differentiate the skunk scat from the fox scat, because as Laura mentioned, the island foxes are everywhere. So there's virtually not many footsteps you can take without stepping on fox scat. Another important part of the training approach was structuring the training so that the dog and the handler could gauge fresh odor versus older odor. If you think in terms of currently occupied dens versus last week or last month, or maybe permanent residence versus vacation home versus visiting a friend. So this information is especially helpful for future habitat mapping of den sites and gathering valuable behavioral data beyond the scat collection. We also factored skunk behavior into our search strategy, and time of day has a great significance for choosing when searches are conducted. The early morning searches are an advantage because skunks have been out foraging at night, returning to the den sites around dawn, leaving behind some fresh, smelly tracks as clues for moose. Evenings are also an advantage because during the day, the ground warms up, and then in the evening, as the air temperature cools, it creates a thermal gradient where, if you can imagine, like in this picture here, the scent plumes of odor are pushing out of the dens, making it more accessible for moose to detect at greater distances. And even though we were there in December, it was still quite warm. And so we also needed to search during these times because they were simply the coolest part of the day for moose. However, these search strategies are important for future planning of work that might take place in cooler climates, because surveying during these times will set the dog team up for the best chance of success. Next slide. We learned about a helpful tool for exploring more during future work. Early on, Moose was telling us about dens, and because many of them had so many twists and turns or were obstructed by debris, we realized we needed a borescope type camera to take a deeper look. So luckily, Lara was able to acquire a borescope with a camera that we could use, or we could figure out how to use. It's a process. This gave us a whole new perspective into the dens, where we saw some neat things that they had collected, and most excitingly, scat. We encountered a few den sites where we could see scat with the borescope, but it was still too deep for us to reach with our hands, and digging around would have been too invasive and compromised the integrity of the den. So what we would hope for in the future is to find the ideal bore scope with sturdy grabbers on the end to be able to collect those scat samples. And in the meantime, I guess we could get some practice runs in with those claw machines, you know, the ones where you try to grab a stuffed animal or prize and hang on to it for dear life before you spend your life savings at the machine. We also learned how important it is to stay on top of safety, to continue explore options for protection against foxtails. Foxtails are the seed-bearing parts of certain grasses that are prevalent on the island, and they can be hazardous to dogs as the barbed seeds can burrow into skin, eyes, and ears. As you can see, there's one pictured here that hitchhiked its way into Moose's ear and landed us a surprise trip to the mainland for an emergency vet visit. Thankfully, it was a quick and smooth removal process, and Moose was happy to get back to sniffing. Next slide. This is a picture taken with the borescope of a skunk scat in a den. You can see the piece directly in front of you, but if you look over to the left side, you can see where there's another piece as well. Again, the scope was a pivotal tool that allowed us to not only take snapshots, but videos beyond what we could see. Next slide. Some of the project outcomes, our first day, which was also Moose's first experience on a really bouncy boat ride. We arrived on the island, unpacked, and Moose hit the ground running, finding his first occupied skunk den within 20 minutes. Luckily for all of us, we didn't get sprayed, but the skunk did let us know that we were close to its home by dropping an odor bomb, which was a welcomed confirmation that Moose was well prepared for his assignment. Over the 21 days that we were there, Moose located 16 den sites. We collected 11 scat samples, and there were a few additional samples that we weren't able to reach that were deep inside the dens. Lara and her team were able to practice visually identifying some of the unique differences between skunk and fox scats, thanks to Moose confirming that they actually were from skunks. And there may have been a few instances where certain humans that will remain nameless couldn't resist satisfying their curiosity to test their human sniffing skills on the scat samples. And next slide, please. So here I will pass it back to Laura to share details about the testing results and looking ahead into the future. I look forward to any questions that you might have.
Thanks, Mark. Yeah, so like Mark said, Moose alerted us to 11 unique skunk scat samples, and we wanted to confirm his ability to distinguish between fox and skunk scat by sending these samples off for genetic species confirmation and also eDNA metabarcoding, so looking at the DNA of all the prey animals that might have been eaten by the fox or the skunk that was the owner of the sample we collected. So in addition to the 11 skunk scats, we collected an equivalent number of fox scats from the same area and sent them all out for testing. And the testing confirmed that Moose is really accurate at finding skunk dens and scats, which was not super surprising, but it was a nice confirmation. So 9 out of the 11 scats were confirmed to be from spotted skunks. One of those, they just couldn't amplify any usable DNA from. It probably also was a spotted skunk, but might have just been too old. And one turned out to be a fox scat. And we were able to successfully amplify DNA from different prey items in the scat, even though the skunk scats were all and fox scats were all of indeterminate age. Some of the most common prey items in the skunk scat included deer mice Jerusalem crickets or potato bugs, which are these crazy baby alien looking bugs that burrow in soil on Santa Cruz Island. And most surprisingly, we got storm petrel DNA and skunk scat that was on the west end of the island near some cliffs where storm petrels are known to have colonies, which is pretty unique result for a terrestrial carnivore. Next slide. So like Mark mentioned, we were only on the island for 21 days. This was very much a pilot study. Detection dogs need a fair amount of downtime, but given the intensity and nature of their task, they can work for a limited amount of hours per day. So given all that, it was a really successful pilot study. It was a proof of concept of this methodology, which had never been tried before on the islands. We were able to locate dens at kind of an equivalent pace as what we would do with, radio collared animals, which is really exciting because this is completely non-invasive to the skunks and it allows us a lot of flexibility in how we work. So for the next steps, we've written up the results of this report and we would like to expand it further. There's a lot of opportunities with detection dogs on the island. The first would be just continuing to map out the network of den sites on the island. So looking at opportunities to put cameras on dens so we can look at fox skunk interactions, look at where skunks are choosing to have their dens, and then reproduction. Very little is known about when skunks give birth, how many offspring they typically have, what's the survival rate of those offspring. And so any information we can learn about their dens is going to contribute to our understanding of the species. Obviously, further expanded diet analysis across seasons and across the island will be really helpful in understanding how foxes and skunks partition that niche and how they choose their prey items and how that changes with environmental conditions. You can use SCAT to look at disease and parasite prevalence. And then finally, the golden ticket would be genetic identification of individuals using SCAT. We haven't had a chance to test this yet with the SCATs we collected, but it is theoretically possible to look at microsatellites in each SCAT and identify the individual that produced it. And so given enough successful replicates, you can potentially get at a population estimate this way, which would be really exciting and is something that we've just struggled with this species for a long time because they're so difficult to detect. So I'm really excited about the potential for this tool. Really impressed with Moose's work ethic and his joie de vivre. It was really fun to see. So I'm hoping we can continue this work in an expanded project in the near future. So I'll pass it back to Crystal to kind of wrap us up here.
All right. Thank you, Lara, and thank you, Mark. Just a little bit more about how you can continue to support some of this work. So we are a nonprofit here at Working Dogs for Conservation. So first and foremost, we love and appreciate any time you can share about us. So I do have our website listed here, and then we are most active on Facebook and Instagram, but also YouTube. We're reactivating our LinkedIn account, and we have just started a Blue Sky account. If you are ever able to donate, that is always, always appreciated. We can take many different forms of donation, including donor advised funds, which is the DAF there, and stocks. We would love to talk with you about planned giving or estate gifts. And then I do have to highlight, really cool, we just started a vehicle donation program. So any type of vehicle, whether that be a car, trailer, boat, even an airplane, can get donated to WD4C. And so it's a third party that sells that vehicle. They can even tow it away for you at no charge. We get the proceeds of those sales and you get a tax deductible receipt. We also have an Amazon wish list, and all of that is on our website, and we can share that with you as well under our Ways to Give tab. Also, if you're in the Montana area, we'd love to have you attend any of our local events. And then the event that I'm highlighting today is the Canine Classic. It's our biggest fundraising event of the year. It's at the end of August. Registration is open now. There's a dinner in the evening, and then the run is the actual Canine Classic. There's a half marathon, a 5 mile, and a 2 mile, and it's a run or a hike. Super fun, dog friendly. So imagine the biggest dog party of the year is going to be with us, and we have live dog demos with our conservation dogs. Amazing food, and it's all at the Paws Up Resort. So just a little invite. And if you can attend it here in Montana. Otherwise, we have remote and virtual options there as well. And then just a huge thank you to all our partners and collaborators. So of course, the Nature Conservancy, Friends of the Island Fox, California Institute of Environmental Studies, Santa Barbara Zoo, Turtle Bay Exploration Park, Sequoia Park Zoo, UC Davis, Island Packers, and the National Park Service. And to all of our amazing supporters, those of you who are joining us today and watching this later, thank you, thank you, thank you. And so I'm going to open it up now to questions. So if you do have any questions, please put them into the chat, and then I'll start reading them off. And Mark and Laura, you can decide who answers them. So first question that came in is, how long did it take to train moose to master the target scent of skunk?
I'll take this one. That's a great question. So Moose is one of our organization dogs, and I mentioned that this was his first deployment project. And what that means is Moose had spent multiple months at our headquarters in Montana, and here's where he learned sort of the rules of the game on a neutral target scent. So he kind of learned the context of, I'm looking for something, I find something, I tell you about it, and then we have a big party. So he had all those rules down pat over the course of multiple months. And for this project, because it was a pilot, part of the pilot was to do the training on the island while we were there. So Moose started his training about 2 to 3 weeks before we deployed to the island, and then we can continue training while we were there for those 3 weeks.
Excellent. Next question is, since foxes are notorious for overworking, is it possible that a fox overmarked a skunk scat that quickly degraded and that moose was alerting properly even though a skunk scat was collected?
That's an excellent question, and very possible. And these are things that we see sometimes with working carnivore scats, a lot of overmarking and things. So it is something that happens. And like Laura mentioned, if this was work that we continue to do on a broader scale, then we'd be able to gather more data and incorporate that into training methods. So that's a great question, very possible.
All right. And then another question is, how did you strategize searches? Were you working in transects or just free range or some other search strategy?
I'll take this one too. Yeah, it's another great question. We did a little bit of all of the above just because this was a pilot project. So it was a lot of learning on the ground while we were there. And our strategies also changed depending on the terrain because we were working in a lot of ravines and tight places where the landscape sort of dictated what our transects would be. And then we also wanted to lean into some behavior patterns for the skunks. So prioritizing Moose's efforts and kind of putting him in the right places he needed to be.
Yeah, and I'll also just add that, because this was a pilot study, we wanted to and because Moose was still learning on the job, we wanted to maximize his chances of finding skunks. And so we didn't have, we only had a short amount of time. So we didn't want to do just transects across the landscape, including in areas where we were pretty sure there weren't going to be many skunks. And we really focused his training on areas where we tended to catch more skunks. Or we thought that it would be, relatively easy for moose to move around and just cover more search ground, or all of the above. Santa Cruz Island is super rugged and has, now that the pigs and sheep are gone, it has a lot of dense vegetation. And so that is definitely one challenge for, a dog. Moose is a little dog. He's like 30 pounds, I think, maybe less. But it's still difficult to kind of get stuck in and penetrate that thick vegetation. So we started off just working along the bottoms of drainages where we, again, tend to catch more skunks and traps. And that would kind of concentrate the scent down into the bottom of the ravine. And then also on trails and roads where Moose could kind of like cover a lot of ground and move pretty quickly and then, pick up on a scent and then go off the trail to follow that a little bit deeper. So that was our approach this time. If we were going to do like an island-wide survey, we'd want to come up with some kind of more, consistent transect design. But I do think, given how hard it can be to find skunks, you want to try to maximize Moose's chance of encountering them, however that's possible.
Another question that came in is, what sort of reproductive data would you like to collect?
Yeah, great question. Well, we'd like to know, I think, particularly about spotted skunk kit survival, which is a tough question to ask and answer. But, learning more about maternal dens and what time of year that skunks are reproducing, if there's any, if the females are kind of solo moms or if there's any co-parenting going on, how they choose their dens, how many kits they have. That will all contribute to a potential future population viability analysis. So we can look at how these population parameters are changing over time for skunks and figure out if they're on a trajectory to remain stable or decline to extinction or increase. So that's just always been a big question mark. Another weird thing is that there's a really imbalanced sex ratio in the skunks that we capture. So we catch like anything from 2 to 5 males for every female, and it's unclear if that happens at birth, if that's just an artifact of our trapping. Maybe males are more likely to go into a trap or more likely to trigger the trap. Or if, females have a lower survival rate for some reason. So yeah, that's another big question that would be great to answer about, yeah, their reproduction and birth rates and all that.
That is so fascinating. That's really interesting. So that's something where the scat, once you run it for sex as well, could really show more about that, I'm sure,
Yeah,
right?
great point.
Okay, some more questions have come in. What kind of borescope did you use that you could get through twisty dens, and was it robotic?
Yeah, I can answer that. So it was actually something we had on the island already. After Moose found his first den, we were like, oh my gosh, I wish we had some kind of endoscope or something to look more deeply. And we happened to have one in our machine shop that is used to like investigate like parts of a car that are hard to access, like in the engine block, or else you can put it down plumbing to look at like clogs. So we just borrowed that, and it's not really robotic except at the very end, the tip with the camera can be manipulated up and down, so that's kind of helpful. But yeah, it's a little bit challenging. You have to bend the arm in the direction you want to go. So if anyone knows of a version of that that has some pinchers on the end, I've been looking and I haven't found anything yet, so we might have to get a robotics class at UC Santa Barbara to build one for us or something.
Another question is, has Moose shown any aversion to the skunks or their odor?
I'll take this one. Yeah, good question. That's something that we tested out first thing when Laura was sending us the training samples. We often test that out with most of our target odors just to make sure the dogs do not. And yeah, he did not. I mean, I personally had a little bit of aversion when they would spray. It was quite difficult to manage that odor, but Moose was okay with it.
Excellent. Any other questions? It looks like that's everything in the chat so far. And I'm just looking in our question section. All right. So I'll just highlight that Laura shared islandspotedskunk.com is a resource. And then if you have any other questions, I do have our info listed up here as well. If you just want to send it to info@wd4c.org, we can direct it to whomever you need to reach, and we're happy to answer those questions as well. So thank you everyone so much for attending. We've had a lovely time. This has been recorded, so we'll send it out as well to everyone who's registered. So if you're here today, you registered, and then if you're watching on the recording. But thank you everyone for coming, and we hope you had a good time, and we hope you like skunks even more now. All right, bye.
Thanks, everybody.